we will start
there. So, and that's the arrows, and there's a pointer.
Half of it is oxidation, basically. Half of it is oxidation. You can drive that half of it with a low potential reaction. Right. You don't have to have right. And then the second half happens. Yes. Right. So it's very electrons go off.
Okay, so our second morning session is on carbon fixation, and this is a deeply connected. Okay, so please
Everyone, please sit down and finish your conversations. Thank you.
So,
Okay, did we are starting the next session. Thank you.
yes, carbon fixation is deeply connected to the evolution of life and also surface life signature, surface environment. So, and in this session, we will have 5 speakers, and then we will begin the first keynote presentation by the. Yes. Oh.
Yes. Everybody hear me? Okay. Yeah, so hello everybody. My name is Bo. I'm currently a postdoc in the Jewett Lab in Stanford University. We do cell-free systems, synthetic biology, very far from me from what you guys are doing here. I have a background in carbon fixation, though. I come from Toby Erb's lab. We're also kind of jumping in a bit for him here. Yeah, for me, photosynthesis or extraterrestrial photosynthesis is the carbon fixation aspect, and we have heard a lot about all the light reactions part of it. So now we'll delve a bit deeper into the so-called dark reactions. And with the next slide, you will see what I thought about hearing extraterrestrial. And I always have a kind of more applied sense in mind. So in this case, maybe take it with a grain of salt that our pathways and potential solutions to improving carbon fixation might also inform what we could expect to find on another planet, given that we have some kind of way to detect it. But I thought about it in this way, and I want to give solutions for these 2 problems. If we ever want to go to another planet, to another world, we need to survive while going there. And ideally, we also feed and have food available. And for this, we probably need to fix or recycle carbon on the way there. And once we're there, we, of course, ideally at some point want to terraform it, or at least want to fix carbon to sustain life. Because for me, that is a big part of what it is. And now I will give you a brief overview of how nature kind of solved the carbon fixation problem, and then what synthetic biology solutions we have available to go a bit beyond. So we don't really need this slide here for anybody in the room. I just find photosynthesis quite fascinating. For me, it's very interesting to see that you can evolve a biological process that can literally burn water and derive the electrons from that and use the abundant available energy source to then proliferate all across. But from a synthetic biology perspective, for me, what it really is in its simplest form, I like to divide things into digestible modules. And for me, photosynthesis is effectively an energy module, and it's a carbon fixation module. And I'm not so sure how many people here are aware of the diversity on the carbon fixation side. We heard a lot about different ways how we can harness light energy, but there's also quite a few different ways how we can fix CO2. And in fact, by now we have found 7, maybe 8 CO2 fixation pathways in nature. And I quickly want to go over them. And I'm using a plot derived from Charlie Cotton's review article here from the time in the Evan lab, where they looked at different carboxylases and plotted their catalytic efficiency on the y-axis over the turnover rate on the x-axis. And what we see is when we look at the classical Calvin cycle, that Rubisco is not the best enzyme. It's not terribly fast. It's in fact more of a moderately efficient enzyme. And besides the reductive TCA cycle, basically the slowest known carbon fixation pathway on the planet, all of the other pathways, such as the 3-hydroxypropionate bicycle and the 3-hydroxypropionate 4-hydroxybutyrate cycle and the decarboxylate 4-hydroxybutyrate cycle, which all employ acetyl-CoA and propionyl-CoA carboxylases, which are biotin dependent and very fast. The Woodley-Jungdahl pathway and the reductive glycine pathway, which molybdenum or metal-dependent format dehydrogenases, which are also very fast carboxylases or CO2 reductases, all of which are superior, at least in terms of kinetic rates, to Rubisco. And so it's quite interesting to me that during history, we only have evidence of these 3 events where these carbon fixation modules were combined with any form of photosynthesis. And maybe I will also echo of what Anthony was saying, that this might be a priority effect, that only the carbon cycle was ever combined with this crazy innovation of using water as an electron donor and hence could dominate everything. Okay, so for now, photosynthesis effectively is the carbon cycle. That is the main dominant carbon sink that we have, and it supplies all of our food, meaning that essentially all carbons in our bodies once pass the active site of this enzyme right here. So ribulose 1,5-bisphosphate carboxylase oxygenate, shows Rubisco, is the dominant enzyme of carbon fixation on the planet. In fact, the biomass of this enzyme alone probably exceeds all human biomass by a factor of 10. However, another aspect of why it's so abundant is that it is a faulty catalyst. So of all the carboxylase diversity I showed in different pathways, this is the only enzyme that also performs an oxygenation site activity. And that's very harmful. So normally we carboxylate ribulose bisphosphate to 2,3-phosphoglycerate molecules, but we can also oxygenate and form a 2-phosphoglycolate molecule along with another 3-phosphoglycerate. And this 2-phosphoglycolate is inhibitory to metabolism. It's a wasteful side product, and it's a loss of carbon if you don't refunnel it into the carbon cycle, and hence it has to be salvaged. And this process of salvaging it is known as photorespiration, at least in plants. In bacteria, we by now call it 2-phosphoglycolate salvage, because in many bacteria that are not photoautotrophic, we also have the carbon cycle. And yeah, so basically that is an issue that nature now had to fix, this oxygenation inherited 2-phosphoglycolate formation. Nature mainly does this by essentially limiting oxygenation flux by increasing the available CO2 Rubisco Cs, and this is known as a carbon concentrating mechanism, and there are several solutions to this problem. So I quickly go over the C4 and crustacean C and acid metabolism aspect of it, which in a very simple way show how evolution worked. So here we have essentially a mini cycle of one of the, if not the most efficient carboxylase on the planet, PEP carboxylase, fixing bicarbonate, forming oxaloacetate, reducing that to malate, storing the malate and shuttling it, either spatially separating it for C4, temporally separating it for CAM, and then decarboxylating this malate near Rubisco, giving it higher CO2 concentrations locally, allowing it to function better. And my first mentor and the person who inspired me to look into carbon fixation, Aaron Bevan, always called this a good example of that evolution is a tinkerer. And that we as synthetic biologists or as engineers could try to, for instance, build a cycle based on PEP carboxylase, not perform this wasteful carboxylation and decarboxylation. And such proposals he also published in his 2010 paper, and people are working on this. In fact, what Rubisco, or what evolution decided to do with Rubisco, and I'm filling in a bit for Toby's lab here, who is focusing on Rubisco a lot lately, Rubisco was evolved to complexify. What we see on the right here is a phylogenetic tree of Rubisco. There are various different forms, and I think we also had them on the posters, which are very nice. I just quickly want to highlight what we have in terms of Rubisco diversity. Essentially, all Rubiscos are dimers at the catalytic core, homodimers of antiparallel subunits, large subunits, and probably the most ancient form are the Rubisco-like proteins or the form 4s, but that's highly debated. Those are non-carboxylating. Everything else is carboxylating, but maybe not relevant for the carbon cycle. So form 3 usually are not relevant for the carbon cycle, but they perform usually also carboxylation of ribulose bisphosphate. Then the form 2s are usually faster Rubiscos that are less specific and mainly bacterial. And then we go to the complex I'm showing here in a little schematic that looks like basically a barrel structure of an octamer of large subunits capped by 4 small subunits from the top and small subunits from the bottom. That is a form 1 Rubisco that are dominant in all real phototrophic organisms, such as cyanobacteria and higher plants. And in the 2022 study from the Air Lab, they were also able to investigate why this small subunit now came to be and why Rubisco became kind of addicted to this. And they were able to use, again, this ancestral sequence reconstruction to resurrect ancient rubiscos and resolve the time point where you had an octameric rubisco that did not yet need the small subunits, and then got it to the point where it was able to interact with small subunit. And there they were able to show in this data here that seemingly gave this rubisco with the small subunit a 10-post higher temperature stability. And upon another substitution, they could show that with this extra substitution, docking of the small subunit gave it an allosteric improvement, roughly a 2-fold specificity increase, potentially showing you the lineage of how the small subunit was recruited. And then in contrast to this, recently they have pre-printed an article where they now also found a rubisco derived from a small subunit already addicted clade, which now lost its ability or its need to interact with a small subunit while still having it in the operon. So this rubisco you can essentially express with or without, and you get different kinetic performances. And as you can see from the top graph here, you can then also titrate in the small subunit again and not only get a specificity increase, but also a huge rate increase. So that's kind of interesting because previously people had assumed that you would become entrenched and addicted and be in need of further complexification and couldn't go back. But now they kind of have to reassess what exactly happened here to rubisco. And yeah, this further complexification is evident in plant rubiscos. So here on the left, we see this ancestral, more bacterial pathway where essentially you just need the giant chaperone and grow ears and Gruyere, and that is mimicked in plants. But there you have additional chaperones coming in, such as RUF1, RUF2, and BSD2. And these chaperones then essentially allow plant rubiscos to fold their form one rubisco, or plants to fold their form one rubisco. And there the question was, they didn't really come with more specificity increases. So how required are these chaperones really for this type of rubisco? And there the Hochberg lab, which is also in Marburg, which is also has been involved in many of these rubisco studies, used again ancestral sequence reconstruction to kind of elucidate an ancestor which still had all of these chaperone requirements, but then you can use substitutions to relieve them of these chaperones and get the same kinetic ability as the non-chaperone requiring rubiscos, showing that also this seems to be an interesting fact that plants decided to, or that they have complexified that much without any evidence on the kinetics parameters, potentially reflecting a regulatory or so need for these. And then in terms of carbon concentrating mechanisms, I think we'll have a talk after this session as well on this, so I'll just quickly highlight it. Obviously, there are very intuitive carbon concentrating mechanisms by encapsulation of rubisco in a proteinaceous shell or in a liquid-liquid phase condensated, separated condensate, where you could pump in bicarbonate and have a carbonic anhydrase in there, convert the bicarbonate to CO2, thereby creating a higher CO2 atmosphere. But all of these solutions, for me, they only augment the core issues of the carbon cycle. And we don't have to talk about in this audience how also the light part of the action is could be improved. So overall, basically only a minuscule fraction of light energy is actually retained in the chemical bonds after photosynthesis has taken place. And we could find a potential better solution using physical-chemical means. And what this is for me is artificial photosynthesis. So if we can separate light and dark reactions, we can potentially get the most efficiency out of the entire process. So already, I think, what was it, 15 years ago, photovoltaics were proposed to be much more efficient at capturing light energy. There, I think the proposal was to make a hydrogen from electrolysis of water, but we can also reduce CO2 directly with a bit of deficiency. But we can then store this electricity energy in formate, which is, at least was a long time for us, considered a very nice mediator because it's quite stable. It's not very toxic, it's permeable, not volatile, can have has a low reduction potential, and has very fast energy reducing enzymes available, which we need to build biomass. And then these physical chemical means are quite good at this, but to make more complex molecules, I'm losing my mic, to make more complex molecules, biology usually is much better. And here, then you have natural C1 utilizing organisms that can then eat the formate and make value-added chemicals from that. However, also for these bugs, most of them use the Calvin cycle for metabolism of formate, and that is again coming with the same bottlenecks that we already discussed with rubisco. But even with this setup, a colleague of us has recently shown 5 years ago that given the same limited land area, I think this is what I would also translate for potential space exploration, where we have limited space on a spaceship. You would have a comparison here of a soybean field making a bunch of protein to feed about 40 people, and you could have a photovoltaic hybrid photosynthesis setup with direct air capture and microbial cultivation producing about 10 times the amount of protein. So this artificial photosynthesis approach, at least for protein production, can be vastly superior. And now I want to tell you all how we can use synthetic metabolism to go even a few steps beyond. And now we're talking format assimilation. We've already covered that it might be wasteful to re-oxidize the previously captured CO2 in terms of formate. We can use a synthetic pathway. This pathway was invented by Aaron Evan in his 2012 paper. It's called the reductive glycine pathway. I've already shown you that we also have that now found in nature. We found in 2020. And that might be a good indication of that some of these synthetic designs we might also at some point find in nature. So this pathway operates as such. Formate is first activated and reduced and dehydrated to methylene THF, which then goes into the name-giving reaction sequence of the glycine cleavage system. But in this case, we use it in the reductive direction, forming glycine. And glycine can then be condensed with another methylene THF to serine, which can be deaminated to pyruvate, from which we can make all other biomass precursors. And from here, and then we also have a formate dehydrogenase to power the system. And from here, then we can achieve formatotrophy. So I said this pathway should be the most efficient formate assimilation pathway. If we compare it to the Calvin cycle, which costs 7 ATP per pyruvate, this pathway only costs 2. So that sounds like a fairly big increase. However, when you factor into account amino acids, polysynthesis, a few other processes, you get to a predicted yield increase of about 20%. Okay. And now we chose an organism to perform a test in where we actually wanted to show that this pathway can indeed achieve this performance, which has never been done before. And here we used cupriovidus necator, which is for me one of these aspects of carbon fixation that has been, basically it has been missing from me at this workshop so far. So I always think about other means of essentially lithoautotrophy, where we can use hydrogen, for instance, and CO2 and form biomass this way, completely decoupled from light. And this is what this organism can do. It's also fairly genetically accessible, has one of the fastest known formate dehydrogenases known, and it's also regarded as safe as in food and feed as dead cells by the European Food and Safety Authority. The next few slides will be quite constant. I will show you on the left the schematic of the cell, what I've been doing to it. In the middle, I'm showing the carbon fixation pathway being active. On the right, I'm showing you bacterial growth curves. Optical density increases over time, and the color should be matching. So in this case, the black Calvin cycle strain is growing via the Calvin cycle. When we knock out Robisco, that's the brown line. And now my kind of direct predecessor, Nico Claessens, had already expressed this reductive glycine pathway, forming a 2-plasmid system in the strain. However, the strain's performance was not as expected. The strain grew quite slow, and the biomass yield was low. This is kind of my PhD project and where I came in. And I decided to basically improve expression levels and at the same time remove plasmid burden. And to do this, I cued the strain of the first plasmid. Now with this activation module missing, which was actually derived from a serine cycle user, the strain cannot grow on formate anymore, as you can see. And if we then provide this module again, but this time with a lateral gene transfer with a transposon approach, we could now select for optimal activity of this pathway, form the trick one more time to then insert the entire reductive glycine pathway into the genome. And what we now have is a fully synthetic way to grow on formate. So no organism ever has grown on formate that we know of on formate using exactly this reaction sequence. It's also different than the previously mentioned CO2 fixation pathway of the reductive glycine pathway in some aspects. And now to accurately assess the biomass yield of these strains, since they had different growth rates, we wanted to use a bioreactor process called a chemostat. And what this machine does here, it provides a constant influx of media with nutrients and efflux. And over time, due to the limited timely availability of nutrients, the cell's growth rate equilibrates with this dilution rate. And hence, we can force two differently growing organisms to grow at the same growth rate. And we compared this then to a similar experiment these are our model predictions, and those we compared to a similar experiment by the Schoonwald et al. Study, where they showed about 4 grams per mole formate of this organism using the Calvin cycle. And our Calvin cycle using wild type came pretty close to that. And now for the final experiment, we now looked at the yield of our synthetic strain. And here we saw for the first time that we can use synthetic metabolism to exceed nature, to exceed at least the Calvin cycle for growth and formate. And so with this, I've shown you that we can use such a hybrid artificial photosynthesis approach coupled with synthetic metabolism upgrades to feed way more people than the same resource area could supply in terms of a plant-based approach. Now, what do we do with our atmosphere? And as we've learned, there's a bunch of them around with very high CO2 and very high pressure, which makes a bunch of the pathways we've been working on actually quite a bit more favorable. In fact, I want to drop a note here. The Wood-Ljungdahl pathway is favorable and even ATP generating because it can exploit a very narrow niche. That niche can be expanded if we have way more CO2 available and way higher pressure. So it might even be that we find different other means of energy generating carbon fixation pathway on these other worlds. No idea how we can detect them, but basically it can be quite abundant, I think. Okay, in fact, if we have high CO2, that negates a bunch of the previously mentioned problems with Rubisco. If it does not oxygenate, there is no real issue besides a bit slower kinetic rate of Rubisco itself. But the pathway actually is not too bad. How can we now test, build, use the Calvin cycle for all of the diverse conditions that, and probably way more diverse conditions than the ones I'm presenting here? So how can we do that? And I want to propose to you, which is basically the trajectory I've been on for the past 8 years. So I've performed my undergrad in plant physiology, moved to microbial engineering, and now I stepped into cell-free systems. So what I want to propose is, what if we take what makes the plant and what makes the Calvin cycle? We don't work in the plant because it takes decades to engineer many things. Even in bacteria, my last project was the work of basically 12 years. What if we go even simpler? What if we go to an in vitro context and build the Calvin cycle there? And that is kind of my current postdoc project. And we can use cell-free protein synthesis to achieve this. And we can use pathway prototyping to explore 100,000 or if not even millions of combinations of enzymes to explore all kinds of pathway architectures and performances across conditions. So what the Jewett lab is famous for and what they do all the time is this process known as cell-free protein synthesis. And what they do here is they make crude E. coli extracts, the S30 extract, but by now we're doing S12 extracts. You supplement what's a bit needed for making immense amount of protein, such as amino acids, energy, a few other cofactors. You can then aliquot that as many times as you like. You supplement per aliquot a template of your interest. And those then, over the next 4 to 20 hours, enriched lysates, heavily enriched lysates with one single protein can then be combinatorically mixed and read out to screen all kinds of pathway or enzyme combinations that you would be interested in. So I want to use this technique to then plug in different Rubisco forms, different other Calvin cycle enzymes, and explore essentially the fastest possible Calvin cycle, the most energy efficient Calvin cycle, maybe even the most cold tolerant Calvin cycle, all kinds of ideas. Now, how do we actually do this? So I started in February, so we are still a bit in the early phase, but the lab I'm from, Toby Erb, is quite famous for building in vitro cycles. He did this in 2016 with this famous catch cycle. This was a synthetic pathway, but in my case, I want to build the Calvin cycle. And how did they do it here? They essentially just took a linear segment of this potential pathway that they could read out. In this case, for instance, I could use the glyceraldehyde 3-phosphate dehydrogenase reaction and read out an NADPH depletion assay. And then I would extend that upstream further and further until I have a full reaction sequence where potentially my substrate is already my product, hence closing the cycle. And this is exactly what I set out to do. So I'm showing you the schematic of the Calvin cycle on the left, which how many enzymes are active in sequence, shown in green. And on the right, I'm showing you now that I can perform a Rubisco assay, which is essentially coupling Rubisco, phosphoglycerate kinase, and glyceraldehyde 3-phosphate dehydrogenase, reading out NADH depletion. And what the value here is essentially normalized to milligram pathway protein, total protein I've invested. So that works, that's great. Now I can sequentially try to extend that, purify more protein, spike it into the reaction mixture. And I did this here for the intermediate xylulose 5-phosphate, which is part of the Calvin cycle. I'm also labeling it under the bar chart so you know where we are. And that has worked, although we got some rate decreases. Now I'm extending it a bit further as well to ribose 5-phosphate and putting in the ribose 5-phosphate isomerase. That has worked. And now the plan would be, okay, let's skip through the entire pentose phosphate pathway and the fructose bisphosphate steps and try to see if we supply GAP, glyceraldehyde 3-phosphate, and we supply abundant amounts of NADH, the only real way it could go is one full turn of the cycle, deplete NADH per GAP, and then we would get a readout only if we perform one full cycle. And this has worked for us, and so this is the first evidence that the cycle can be functional in vitro. We are also able to improve this a bit by feeding more GAP, which then decreased a bit, feeding even more GAP, which already shows us how important it will be that we have access to this prototyping platform based on lysates to really explore all kinds of pathway solutions. I also want to highlight here for transparency's sake that this just shows really one turn. This doesn't represent an in vitro autotrophic system. What it needs is continuous energy supply and basically needs to fix more CO2 per acceptor to really be called an autotrophic system or whatever that means on a cell-free equivalent. So now next, Rubisco is, as I said, still maybe a bottleneck in terms of its kinetic rate. And we didn't discuss this so far, but essentially Rubisco is locked in an interesting trade-off, where essentially the more specific a Rubisco gets, the slower it gets, the faster it gets, the less specific it gets. And these natural Rubisco forms that we already talked about kind of represent this. So the fast form 2 Rubiscos are typically also the ones with the highest Km values, apparent Km values, and also the lowest specificity. I would pick one of these Rubiscos now and ideally engineer to be even faster, because assuming we are in a Martian atmosphere or in another atmosphere with very high CO2 concentrations, I wouldn't care about these potential specificity decreases. I could go as much as possible into rate. And I want to propose another method I've been using most of my scientific life, which we can use to, in ultra high throughput, select for this, and that is growth coupled selection. What we do here is we take a wild type strain of E. coli mainly, we perform rational gene deletions to make it oxotrophic for certain metabolites, for instance, 3-phosphoglycerate, and then we provide our Rubisco into the strain to couple activity of Rubisco to growth of the strain, and hence when the strain gets better at growing, usually Rubisco gets better as well. Now to hit you with a bit of metabolism towards the end of the presentation, on the left we have a schematic of the pentose phosphate pathway and glycolysis. On the right we have another growth curve, this time of E. coli growing on glycerol, and I'm showing you how that looks. I've now even simplified the pentose phosphate pathway a bit more. 2 fructose 6-phosphates and 1 GAP make you 2 xylulose 5-phosphates and 1 ribose 5-phosphate. If we now delete one of the enzymes of the pentose phosphate pathway, in this case the isomerase, we get no growth on glycerol anymore. And now we can use heterologous expression to provide the phosphoribalokinase, which is needed to make Rubisco's substrate and a Rubisco, this a form 2 Rubisco, and we can see that we can get growth in this condition. And if we now switch the condition to something even more demanding, so the selection here is essentially removal of xylulose 5-phosphate, and here all of the xylose has to be converted through the synthetic shunt for growth, we now see that the growth stalled or we don't have growth, making a perfect selection space to evolve even faster Rubiscos, which we can also combine with a suite of hypermutagenesis systems that have been published in recent times. I also just want to highlight that we have different selection available to do different things, spanning 2 orders of magnitudes. Not super important, I'm done at this point. We can then take that faster Rubisco, put it into the cycle, and get the fastest carbon fixation cycle possible, which might inform, and all of this combined, all of these combinatorics hopefully will combine to inform us what could be used and could be possible for varying conditions in different worlds. So with this, I hope I could convince you that we can use synthetic biology and especially synthetic metabolism to go a bit further than what nature has shown, maybe even highlighting what already might exist in other worlds. Yeah, with this, I would like to thank all my mentors, especially students that have worked on the Rubisco project, and Toby and Mike Jewett, and you all for your attention.
Thank you very much. We have time for 1 or 2 questions. Brief questions.
Thank you so much for the very interesting talk. I just have like one quick question. What do you think may have preventive nature from optimizing the cycle versus like to what you're showing could be like its optimal state?
Yeah, so I guess it's optimal per condition. I think there just wasn't there's a bunch of, like, these questions usually come up for some of the pathways we present that are synthetic, or in this case might also be synthetic. They usually exploit thermodynamics in such a way that you just push so much more CO2 into it that we can be a bit cheaper or faster in another way, and that is always condition dependent. And so it seems there's no niche here where we can exploit that. Yeah.
Okay, so thank you very much. Okay, let's speak again. Let's speak. Next talk is another 30-minute key talk by Helena from Stanford University.
And use arrows to
Hello everyone, I hope you're enjoying the workshop so far. I will continue the metabolism trajectory here, and especially the carbon fixation trajectory. And I want to take this opportunity to talk a bit more about synthetic CO2 fixation and how we can more efficiently engineer it. So this is work that I did during my PhD in the laboratory of Tobias Erb at the Max Planck Institute for Terrestrial Microbiology in Marburg. I'm now a postdoc in the Druid Lab in Stanford, and I'm really excited to participate here. I've learned so much already, and I think we can all agree that, at least here on Earth, microbes are the terraformers we have. So to put it in very simple terms, life would not be possible without microbes. They fix about 70 gigatons of carbon per year. They make nitrogen accessible to other biological systems, and they can mineralize organic phosphate. And in short, they are absolutely everywhere. So they are extremely abundant. They are extremely diverse, and we can harness that diversity to design essential metabolism. And they are essential for our life. So given their relevance for life on Earth, we can, of course, ask whether we could use these to terraform planets. And I think thinking about such a question, we also automatically get to thinking about which conditions would allow which forms of life. Now, this is not a novel idea, and we now here see a name again that apparently everybody in the room knows, as I heard from Bob's presentation on day one. But Carl Sagan already proposed to use algae to terraform Venus, for example. And this is not my expertise whatsoever, but what I could think of here was that if we have potentially limiting conditions, then we want the metabolism of microbes to be as efficient as possible. Now, in the context of photosynthesis, we know what the bottleneck of the CO2 fixation part is. And it is that the core enzyme of photosynthesis, rubisco, is on the one hand a comparably slow catalyst. And beyond that, while it's supposed to use CO2 as a substrate, on average, every fifth reaction, rubisco uses oxygen instead, which means an efficiency loss for natural photosynthesis. Now, depending on the atmosphere, that might be more or less of a problem, but I want to introduce a concept to you that people in the field, like Tobias Erb, like Arun Baeven, have come up with decades ago already, and that is to use other carboxylases we have a plethora of that are much faster than rubisco and much more specific. And we want to use these to now design efficient synthetic CO2 fixation, which means incorporating these catalysts into a suitable metabolic architecture. Now, this synthetic CO2 fixation metabolism could, if you only look at the rates of the limiting enzymes, in principle allow much higher efficiencies than natural CO2 fixation. It is more energy efficient, it is more specific, and operates faster. Now, what we are working on is to integrate this metabolism into microbes to facilitate the assimilation of 1 carbon units and conversion of them into either value-added products or biomass. And if you want to think about space exploration, you could think about using microbes to recycle 1 carbon units and produce feedstocks from that. Here on Earth, we are primarily interested in this scenario because it could be the fundament of a circular bioeconomy. It could enhance plant biomass yields, but the underlying metabolism is difficult to engineer. So we here envision a stepwise transfer of designs from cell-free systems that Bo just introduced to you to simple model microbes such as E. coli to only engineer the most efficient designs and difficult to engineer higher organisms. The issue with this vision is that so far the synthetic CO2 fixation doesn't work in vivo whatsoever. So I'm working on achieving this first step in the transfer process, realization of synthetic CO2 fixation in E. coli. And for today, I want to ask the question how we can engineer this metabolism efficiently. And I want to give you an idea of how we go about this, what the limitations of the process are, but also what the potential of synthetic metabolism is in other contexts. And I will focus on the one hand on strain development, giving you one example, and then on pathway implementation, either via adaptive laboratory evolution or via automation and machine learning. Now, the latter part might be interesting if we want to, in a very broad sense, brainstorm about automated engineering scenarios that you might want to think about if you want to send microbes somewhere where you don't know the conditions yet and you want to adjust them to survive in these conditions. Now, at this point, I have to admit that just talking about synthetic CO2 fixation is a severe oversimplification. In fact, there have been so many designs that were theoretically proposed. And I'm now here showing you 4 that people in Toby App's lab have worked on. But what I want you to focus on are the colors, because I colored reoccurring reaction sequences for you. And the key take-home message here is that so many different designs are composed of reoccurring metabolic modules. So the building blocks that we are playing with for CO2 fixation, at least on Earth, reoccur all the time, and we can make multiple pathways from those. So we might have to look for multiple pathways somewhere else too. Now, in principle, we would want to use those to convert E. coli, being a sugar-eating microbe, into one that eats CO2 and an energy source, right? But unfortunately, the cell, as illustrated here by David Goodsell, is incredibly complex. And to be honest, we have zero idea in which ratios we need the catalysts of a synthetic pathway in this complex cell for it to work well. And that is the bottleneck that my research tries to address. Now, we need a very clear readout for functionality if we are working with a complex system. And we achieve this readout by first taking away E. coli's ability to synthesize one part of biomass. And then we want to also break down complex pathways into easy-to-test small modules. And modules should reconstitute the cell's ability to synthesize the missing puzzle piece, the missing biomass precursor molecule. And through that, cell growth becomes a readout for the functionality of the module I'm trying to test. And with this approach, we could then iteratively expand the pathways we are implementing until at some point we have growth on sugar as main substrate, but synthesize the missing essential biomass fraction from CO2 via the synthetic pathway we want to realize. Now, this has been a lot of propositions and no real data shown. And why do I even believe this works? I want to here take the opportunity to highlight the incredible work done by the group of Ron Milot at the Weizmann Institute, because they showed how this approach works to introduce the natural CO2 fixation cycle, the Calvin cycle, into E. coli. And what they did here, as you see on the bottom right, was to take E. coli and feed it with xylose, but also CO2 upon expressing the relevant enzymes for the Calvin cycle. And then they increased the fraction, or they kept the CO2 unlimiting while decreasing the availability of xylose. And through this approach, they managed to obtain an E. coli cell that can now use CO2 as sole feedstock to make its biomass, which is incredible. So with this, we hope that we can harness the same approach to realize synthetic CO2 fixation and prototype the different pathways. And I now want to focus a bit more on how we develop the sensor strains for this, focusing on the key output molecule of these synthetic designs, glyoxylate, which you see reoccurring as cycle product already 3 times only on this slide. And the issue in creating glyoxylate sensors is that the metabolite inherently isn't essential for E. coli. So if you take away E. coli's ability to make this intermediate, it can still make all its essential metabolites too and just grows. So here Enrico Orsi, who is now heading a research group at the DTU in Copenhagen, and me were thinking about how to introduce a connecting node between glyoxylate and essential metabolites to couple growth to its availability and select for glyoxylate formation via synthetic CO2 fixation. Now, I just showed you this image of the incredibly complex cell. So how do we even find which deletions we need to introduce? Luckily, we have models of E. coli's metabolism that we can consult to look for knockouts. Here you see one knockout simulated against the other that would confer a glyoxylate dependency, here indicated by blue color. Now, to give you an example, we here now created a strain that requires glyoxylate to synthesize an entire pool of metabolites, the upper metabolism intermediates. And then I implemented this, and we test the strain in the lab. So here I'm monitoring optical density over time. Optical density gives me an idea of how many cells I have in solution, and it's measured, for those of you who are not biologists, by a plate reader, like a device of this size that takes measurements every 10 minutes, a very simple process. And in short, I don't see growth only with the main substrate, but once I supply glyoxylate, I do see cell growth, indicating that, yes, this is a functional sensor. I now have a very simple readout for the availability of a metabolite in this cell. I wanted to use this to implement synthetic CO2 fixation. And in short, a module from the whole PEG cycle. And once I introduced my enzymes that I wanted to probe, I still saw the same picture, so no growth, except if I supply glyoxylate. And a key take-home message here is that this does not mean that the metabolism is dysfunctional per se, just that I'm asking for a too large fraction of cell metabolites to make from this glyoxylate. And we can now go ahead and ask what would happen if we need to make a smaller fraction of the cell from this metabolite. And in short, then we can show that this pathway works. So in trying to realize synthetic CO2 fixation, we have to consider that this metabolism often initially only supports small fluxes. So for glyoxylate, we addressed this problem by creating a suite of sensor strains, which, as you see by the bottom plot, which shows optical density over maximum glyoxylate concentration, is now suitable to detect the availability of this metabolite over a 3 order of magnitude concentration range. Creating these strains takes time, but luckily, at least for E. coli in the community, we have over 100 designs available that are very well characterized. And now we want to use these to realize synthetic CO2 fixation. And here I have to admit that we can't yet be engineers in the sense that we know how to design the system for it to work. So we have to rely on evolution. And also this was an interesting discussion I had already at the workshop. How quick would evolution proceed in other systems, right? But just for those of you who are not familiar with it at all, every once in a while nature makes a mistake and a cell emerges, shown in green, that learned how to use my metabolism of interest. And then this cell in a certain cultivation regime would take over my population. For synthetic CO2 fixation, there were large parts of it that were functional immediately upon implementing them in vivo, but specifically showing that one key enzyme of it works, crotonal-CoA carboxylase reductase, the little gear wheel that reoccurs across designs, that has been tremendously difficult. And I've focused on making this work. So why has this been difficult? Because the reaction sequences downstream of this enzyme are incredibly long until you get to a metabolite that E. coli knows how to deal with. So you have to functionalize many enzymes at once to make this work. And in an attempt to do this, I created the suitable sensor strains for it, for these output metabolites. But for the sake of this talk, I will now only show you one example of engineering a short section of one of these modules in a strain that requires the output acetyl-CoA for growth. And you don't need to know anything about specifically this module. This is an example for how we try to engineer and functionalize metabolism. Now, if I introduce my pathway or my pathway enzymes, I see growth with my positive control substrate and no growth with my pathway substrate. So I ran into the exact same issue we so far have every single time in trying to realize synthetic C1 assimilation. The metabolism does not work initially. I conducted adaptive laboratory evolution too. It took me about 2 years to obtain a strain that grew on my pathway of interest. And I could now go ahead and ask what makes my cell grow. And again, what I found here is something that we also see reoccurring in the field. We have mutations with very broad effects in the cell on a regulatory level that affect so many different things, and analyzing, finding out what makes this pathway work now takes a lot of time. In my case, I found that one of my pathway enzymes had been upregulated. So I want to take the time to introduce a different visualization of pathway flux for you, which you see now on the leftmost side of the screen. And what happened was that initially, probably the abundance of this middle enzyme fumarase A was not sufficient for my pathway to work, and evolution fixed that for me, now allowing enough flux to go into my cell. This leads us to ask the question, what happens if we more efficiently probe the solution space for enzyme abundances? And here I want to remind you that in our system, the enzyme abundance modulates pathway flux, but we can read that out through the growth rate of our cells. So I wanted to tune the abundance of enzymes, and then I thought, okay, let's tune the entire system. Let's also tune the abundance of all substrates, which I can read out through optical density biomass formation. And this is a very large solution space already with 5 different parameters that I now have to find an optimum in, which is what machine learning is optimal for. So we came up with a workflow that interfaces a machine learning tool that had already been established with our in vivo system, recommends test conditions that are pipetted by a liquid handling robot. So all I have to do is take out my cells, or my plate from this robot, add my cells in, monitor growth. And what I found was that in the very first round, I immediately found conditions allowing cell growth. So the 2 years of evolution I had to do beforehand were not necessary whatsoever. If I tuned this correctly, I can get growth in 1 workflow round, so I see it after 4 days. And I could iteratively optimize this growth and at the same time learn more about my system. Now, ultimately, we wanted to know, is there an advantage to doing this evolution at all? So we combined this module tuning with evolution, and in that could really break the thresholds of what we could achieve beforehand. Why should you now even think about this? Why does this matter to you? Because if we want to think about C1 assimilation as a means of generating feedstocks in other systems, we have to consider that so far we were relying on years of engineering to realize the metabolism. And this workflow really allowed us to accelerate this engineering. So coming back to the initial introduction, does this now mean that we can use microbes to terraform planets? Well, not at all, to be honest. So you saw that synthetic CO2 fixation is promising as a concept, but we have to learn so much to engineer it quickly. So we need to get better at engineering it. And what I showed you now was, although a machine learning guided workflow, still a workflow that relies on me as the scientist, as initial decision maker of which parameters do I even want to tune, which readout do I define for my system, and so on and so forth. And I have to be there in person to engineer my system. And a trajectory that, to be honest, I didn't hear much about yet so far, but that I was really interested in discussing and that I'm thinking about a lot, is how we can now further outsource the decision-making process and combine automation and agentic systems to get meaningful outputs and to get autonomous decision-making. And in preparing this talk, something that I was excited about was whether we could combine all this into a standalone experimentation platform. So in case you want to think about this with me during the break, I would be very happy. So in conclusion now, I hope that you learned about the concept of synthetic CO2 fixation and that in principle it can exceed the limits of natural systems, but we need to get much better at engineering it because its functionality depends on the one hand on getting correct enzyme expression levels, but also correct strains. Growth coupling as a technique is really powerful and allows us to screen for solutions, especially if we combine it with machine learning and lab automation. Now, thank you very much for bearing with me through this topic that you're not so familiar with, probably. I really appreciate being here. Thank you to the organizers for inviting me. I learned a lot already, and I would be happy to take any questions if you have any.
Thank you very much. We have time for a couple of questions.
Such a relief to see someone speaking the same language as me. I was looking at your growth rates, and for your E. coli to hit an OD of 1, you were looking at 40 plus hours.
Mm-hmm.
Is there a reason you decided to go that route? Because there's a lot of autotrophs that will hit that in a shorter time frame. So I just wondered, are you looking at, because it's a pretty, E. coli is a pretty malleable system to be able to change downstream products. What was the reasoning in the other than its tractability of the E. coli versus taking an autotroph?
That is the reason is its genetic accessibility for initial prototyping, but also, we are good at engineering it quickly and figuring out how to do the engineering better. But so far, I think if we wanted to produce anything from C1, you're probably off better with the actual autotroph, because we also fail completely at integrating, useful energy systems into E. coli, or we're very slow at that because, we saw before the complexity of the photosystems and so on. We don't succeed at all in transferring those. So it's really just that we are able to initially screen in very short engineering timeframes. But realistically, to produce something, use a microbe that already uses your substrate.
I just want to speak about your talk having finished there. What kind of application of this synthetic carbon fixation? I know you allow tested the pathway, in the E. coli. Eventually, I know you allow use E. coli for any kind of application. I know that my experience is biotech inside as an FDA. I'm allowed pass anything to use E. coli to produce anything for human or animal or any consumption. Related to that one, you could consider to put this pathway in the yeast or something like that. That is my question. That is very safe for human consumption, something like that.
Yeah, I think that, so not me personally, I think research interest-wise, my interest is more in making the engineering process more efficient. So I'm going to stick with E. coli, but for sure the people working on these cycles there have been efforts to introduce even simpler C1 assimilation pathways to yeast, which were then less successful even because of more genetic redundancy that made it even slower to implement them, kind of. But yeah, there's a huge area of research to introduce these to microbes that already assimilate useful compounds, or even Ralstonia eutropha that Bo was talking about that you could use as actual feedstock. So these are ongoing efforts. People do this, want to realize this, and just didn't succeed yet.
Yeah, that was a great talk. I really love that. Just a quick question for your MEVIS guided workflow. Do you need metabolomic data for feedback at all? And if so, how many intermediates would you really need to look at? Maybe not the whole suite, but how much would you restrict?
So I did not rely on metabolomics at all here because I have the growth readout. What I did beforehand, though, so the way you tell the story often isn't the way you started it in the lab. So as validation of my pathway working, I did perform 13C labeling analysis in my strains to show that indeed the module works, and what I get as final output is also my pathway indeed. But if you do the selection strain validation thoroughly beforehand, validating that your selection doesn't break over time and that indeed your pathway is what's rate limiting, then you can really only use the growth readout. Hi.
I'm not a specialist, so I couldn't follow, but
That's good.
But I want to know about the machine learning. That why it's so important, because of course machine learning depends on the training set. So how big is the training set that you need to provide to the machine learning?
So there's no pre-training in the sense that I would have to. So what I do is to follow like a Hammersley sampling algorithm to first sample the solution space. Kind of it's maybe a way of random sampling if you want, and then this is your input data set. This is also only 96 conditions, and then you use this to kind of identify where there could be maxima in your solution landscape and try to move towards those. But that is it. So no pre-training in the sense of me generating much data beforehand and then optimizing the algorithm. The algorithm was initially developed to optimize CO2 fixation systems in cell-free. And there you could look at the publication of Amir Pandi, who was a postdoc in Toby Erb's lab, who initially tried different, for example, models and trainings. But what you need now on the application side of things is just one initial round of random testing of, in my case, I decided to go with the 96 conditions.
Okay,
Yeah.
100, which is a very small.
Yeah, 96, do that in triplicates, and that's your starting data set.
But it's different cycles of machine learning.
Yeah, and then you iterate.
iterate.
Exactly,
Okay.
you iterate through that, and by that get closer to the maximum.
I see. Yeah. Thank you.
Maybe one quick question.
Also, because both and Helena's talks are kind of paired. If you want to ask both questions during this period too, feel free. We have several more minutes. Questions?
Thank you. Really cool talk.
Really cool talk.
Thank you.
I was wondering, I'm not familiar with the machine learning algorithms that much, but how much are these models accounting for phylogenetic relationships, in the sense that there might be some enzymes that are more closely related to some than others? So I was wondering if anyone is trying to account for this.
Yeah, so to make it very short, this model does not account for phylogenetics or even what enzymes you're using at all. So all that you look at is input concentrations and one readout for system functionality, which in my case is growth. So concentrations, I mean, because I use an inducible system to titrate my enzyme abundances. This model is not aware of pathway stoichiometry, enzymes, thermodynamics, anything like this. It's just a way for me to quickly identify which growth condition allows best growth. Because I have to, and I only feed in the correlation of growth and inducer concentrations and substrate concentrations. There are, however, I mean, the enzyme design space and also machine learning for enzyme design is absolutely booming right now. And for sure, there you can find models that take into account enzyme performance and try to map that back to enzyme origin. But that's not what I used at all for this optimization.
So we move to the next presentation. Let's thank speaker again. So we will have 3 10-minute presentations. And the first presentator is so Dr. Slati from the University of Arizona. So.
Testing. Cool. So thank you all for being here for my talk. I think I changed the title slightly little bit after all the discussions I've been having. I also deleted all my introduction slides. We don't need that part here. So the key things that I've been like understanding and interested in photosynthesis is that we have a lot of laboratory experiments where we are trying to create novel photosystems that are better suited Rubisco enzymes and all those things that we just saw here. And those systems are being put into cells and we let them grow to see if they can grow under those conditions. And then we have experimental evolutionary biologists who try to evolve different microbes at different environmental conditions. So nature also did all that, and then we have different kinds of species with different photosystems that has evolved over time, and all this together makes the biosphere. So when you're looking at exoplanets, as much as I would love to go back and like travel to the exoplanet and find some rocky or some other species, we are kind of stuck with using telescopes to understand what are the absorption spectra of those planets. And the absorption spectra can tell us information as to the concentration of different gases or molecules on those planetary atmospheres. So my goal here with this talk is to try to link all of this together and provide a framework that we can use to go from all the novel discoveries we are doing in the lab to a scale of biosphere. So to start with, I'm going to explain two different models. So the first one I would like to bring attention is a model that Leles and Levin in the 2023 paper proposed. So this model is a proteome allocation model, and it has a temperature component to it. And all the green compounds, components you see here are the concentration of different metabolites, and all the blue ones are the metabolisms that either produce those metabolites or consume those metabolites and use them for production of ATP and all those things. Now, each of these blue components, they are all enzymatic parameters, right? So they all have these temperature component that is going to change their affinity or the rate at which they function and so on. Now, what happens with temperature is that when temperature increases, these systems, such as say photosystems, the cells can resort to adding extra photosystems to overcompensate the temperature effect, or change the membrane lipid structure, or increase the number of transporters to acquire resources. So with increasing temperature, cells can come up with these kind of novel strategies that can allow it to evolve at higher and higher temperatures. But then the entire proteome for a cell is most often limited by the size of the cell. So it cannot do everything at the same time. So this is where it brings me to this another kind of strategy-based model that Malik et al. Proposed in 2020, which is the yield, acquisition, and stress model. So what this says is that you have a fixed size of the proteome, and based on that fixed size of the proteome, you can either spend your proteome in all the metabolisms associated with growth yield, or with resource acquisition or stress tolerance. Most of the time, a species that is growing at optimum growth temperature is kind of trying to balance out these three different allocations. So how does that work for photosynthesis? In terms of photosynthesis, we can see that all those light reactions, all the photosystems that produce the energy for the cell, and the Rubisco components that fix carbon dioxide into sugars. So those are all a part of resource acquisition. Now, all the energy and the sugars that we get is going to be utilized for producing biomass, and that's the yield that we're going to get. With increase in temperature, things are going to break down, and that's the stress part of this triangle. So as we increase the temperature, there's going to be more stress, more things are going to be breaking down. So the cell eventually has to take down those resources from yield and acquisition and move towards tolerating those high stress. So we're going to combine these two kind of models to have a thermodynamically informed model. So how are we doing that? So I have 50% of all the proteome is allocated for housekeeping genes. So I'm not going to touch that. The cell has it fixed. And the remainder of the 50% is what I allocate for yield, acquisition, and stress. So under no temperature effect, or at least until optimum growth conditions, you have high amounts of yield, proteome allocation towards yield, so the cell is growing at maximum growth rate. The moment temperature effect comes into play, beyond optimum growth temperature, then things are starting to break down. So you need to focus on preparing those things. And then we have these Rubisco effect, for example, the specificity and all these kinetic parameters are also going to start to change. So what did we do with this model? So I took a couple of input traits. So we have the thermodynamically informed input traits. We have the speed, the specificity of Rubisco, the activation energy to carry out those reactions, the energy that is required for denaturing, and then a stability parameter, which I call TD. TD is just nothing but a temperature at which 50% of your entire proteome is decayed. So the assumption is that if a cell is going to die, it's going to die much earlier than the TD. So I take these input traits, calculate those thermodynamic modifiers for estimating the specific productivities of different components, such as how does Rubisco rate and concentration change with temperature? How does ribosomal concentration and rate change with temperature? And reminding you, this is a very simple model that I'm showing here, but this can be increased to more and more complex models. Now, once we have those specific productivities for each of those components, then we can group them into yield, acquisition, and stress budgets. And by doing so, and then looking at the trade-off, remember the triangle I showed you and where the cell is in that triangle, we can come up with outcomes such as how the growth rate is going to change. What is the optimum temperature going to be given these input traits, and what would the maximum growth temperature be given these traits? So what I did is I used a simple Monte Carlo kind of simulation to look at the entire parameter space. So in this case, I have Earth-restricted data sets and then universal or theoretical species data sets. So what I did is I used Flamholt's 2019 paper and Gunman's 2016 paper to get the ranges of each of those traits, and that's what you see in green. And the triangles that you see are basically empirical data. So you can see that the trend is kind of similar, and I also wanted to show that because we know that KCAT and the specificity, they are correlated, and I'm trying to show that I've maintained the correlation with various values of KCAT and specificity. So, and then what I did is I was like, okay, what if I then use, universal values, which is not earthbound, not something that we know for Rubisco traits, but we can go to KCAT as high as 165,000, which is not possible, but I just put it there just to see what happens. And at the bottom is showing you how the Earth-based parameters and the open or universal parameters change. Once I input these, the results I get is basically, you can see the growth rates versus temperatures. And what we see is that, interestingly, at lower temperatures, you can go to a really low growth rates. And as temperature increases, your growth rate kind of stabilizes. And what was really interesting for me in this particular model is that no matter how high I put these values of TD up to like 130 degrees Celsius and KCAT up to 165,000, for some reason, cells die out at 75 degrees Celsius. That's the Tmax that I can achieve. So that can also be the effect of model, but I'm still working on those parts. To look much closer, what is happening is that over time, with temperature, what the model is doing is that it is increasing the yield with temperature, and as it increases the yield, it reaches a maximum value, that is the output, which is the optimum growth temperature. Beyond the temperature, the yield decreases, and I'm just realizing this plot is not super intuitive. The green lines and the blue lines are the composition, like the fractional composition. The black line is the rate, And the rate is basically the concentration or the fractional composition of green line times the rate at which yield changes or with temperature. I'll be happy to talk more about this after this. So what can we do with this is that once we have these thermodynamic traits being translated into traits of growth and temperature, optimum temperature, then we can put them into adaptive dynamic modeling, which are basically evolutionary models to test if some mutants can invade a population or not. So what that would say is that in this case, I have two simple toy models. One is for the TD, which I said is a temperature where around 50% of your proteome has decayed. So what we can see is that these are called pairwise invasibility plot. And what this shows is that at the line, the resident trait and the mutant trait have the same same values. And above that, we can see that above that, we can see that the mutant can invade the resident trait. So where I'm going with this is the conclusion slide, is that using a combination of thermodynamic parameters and evolutionary models, we'll be able to link what is happening at a single cell as to how it can change the population of a species and how that can affect at a planetary scale. So if you are interested in this, I would be super happy to talk about that.
Okay, thank you. If no, we move to the next speaker. Okay. Okay, let's thank our speaker again. Thank you. So next presenter is Dr.
Yeah, I can say mine.
Yes, sorry.
Thank you.
Carolina. Yeah,
Yes.
sorry, Carolina from UC Santa Barbara.
Thank you. Yeah, I'll be keep it.
You can use the arrows to advance.
Okay, should I use this one?
No, yes.
Okay. It's on. Hi everyone, good morning. My name is Carolina Martinez Gutierrez. I'm an assistant professor at UC Santa Barbara, where we work in the early life and microbial evolution lab. And I had to change my title because I didn't realize that I only had 10 minutes. So today I'll be just talking about whether the carboxysome has always been essential for oxygenic photosynthesis. All right, let's talk a little bit about cyanobacteria. We've been hearing about cyanobacteria a lot during this workshop, and why are they so important and so relevant for astrobiology? Well, they are extremely ancient, at least 2.4, 2.2 billion years old. They are extremely abundant, morphologically diverse, and they invented oxygenic photosynthesis, even in eukaryotes, and that's extremely relevant for astrobiology if we are looking for complex life, say. This ecological success, we think, is largely driven by photosynthesis itself, and this metabolism led to dramatic and major planetary events that were very important for the evolution of life. Of course, we are specifically talking about the Great Oxidation event. However, oxygen is also very bad news for cyanobacteria, and in particular, the carbon cycle. So the first speaker was talking a little bit about this. When there is oxygen around, instead of fixing carbon, Rubisco, which is the key enzyme in this metabolic pathway, uses oxygen. And that's really bad because photorespiration happens. There are some good amino acids being produced, but in general, this is very expensive for the cell, right? So we want to minimize that. And people think that the reason why Rubisco reacts with oxygen is that it first evolved under an environment with high CO2 and very or absent concentrations of oxygen. So these are the conditions under which Rubisco first evolved. And of course, conditions have changed dramatically, right? But Rubisco is still dealing with these conditions. Very smart cyanobacteria came up with a strategy to deal with this, and that is the carbon concentration mechanism. And the most important, well, at least for me, the most important part of this mechanism. We have some inorganic carbon transporters, but the coolest part of it is the carboxysome, which I'm showing here with a red circle. And we believe people in general think that the carboxysome is an adaptation to modern atmospheric CO2 and oxygen levels, right? They are both inverted if we think about the conditions under which Rubisco first evolved. So conditions have changed dramatically. So what the carboxysome does, it densely packs Rubisco and the carbon anhydrases inside it. So the concentrations of carbon dioxide go very high, right? So this enhances the carbon fixation process. So it's definitely higher concentrations than what we will find, for instance, in the cytoplasm. So again, essential mechanism for photosynthesis under modern atmospheric conditions. However, we don't know really when the carboxysome first emerged in the history of life on Earth. So common wisdom indicates that it emerged during the Phanerozoic, again, when oxygen went up at very high concentrations, very similar to what we have today, and CO2 decreased dramatically as well. So those are the perfect conditions to have a carboxysome. However, we know that oxygenic cyanobacteria originated before that. We have fossil records, we have the GOE, and we think that it was caused by cyanobacteria. So what happened there? So this is a hypothetical tree of life of cyanobacteria. For those of you that are not very familiar with phylogeny, this is just showing the vertical evolution since the last common ancestor. And if the common wisdom is accurate, that would mean that ancient cyanobacteria did not have the carboxysome, and we are talking about a late independent acquisition, right? So more or less around the Phanerozoic time. However, is this a feasible evolutionary scenario? So that's the question. So I really wanted to know how ancient is the carboxysome and what is its evolutionary origins. So again, this is really important because we need to understand the physiological requirements for oxygenic photosynthesis on Earth and potentially elsewhere. So what we were doing is challenging this idea of late acquisition of the carboxysome and trying to explore whether this carboxysome could have been present in the last common ancestor of oxygenic cyanobacteria. But this would imply that the carboxysome emerged under rich CO2 conditions and very poor oxygen concentrations. So more around the time of the Archean or potentially the Proterozoic. So to explore this question, actually, to be very honest with you, my collaborators and I were interested in the split between beta and alpha cyanobacteria, but this question came later. So on the left side, I'm showing you the tree of life of cyanobacteria, including the non-oxygenic guys, and the non-photosynthetic cynos. And the panel B, which is on the right, is showing the absence of the proteins that are associated with the carboxysome. And what we can see is that indeed, all cyanobacteria, with a very few exceptions of secondary loss, have the carboxysome. These, to some extent, show us that it was very likely present in the last common ancestor of oxygenic cyanobacteria. So this late acquisition hypothesis is not really supported, and we did a small analysis of ancestral reconstruction just to confirm. So this shows a probability that the last common ancestor had this trait. This was done by my graduate student. And indeed, we found that, yes, the last common ancestor of oxygenic cyanobacteria had it. So it could be that actually the carboxysome is as ancient as oxygenic photosynthesis itself, right? However, again, we know that cyanobacteria are very ancient. So if we look at the geological record, the geochemical record more precisely, we know there was oxygen a little bit around that, well, very dramatic concentrations relative to the past, but CO2 was very, very high. So why would cyanobacteria get a carboxysome when apparently it was not needed, right? So it might have to do more like with oxygen rather than CO2. So this ratio of CO2 and oxygen seems to be the most important part. We were also interested in the how. How did cyanobacteria acquire this trait? And it's very important to mention that the carboxysome is also found in other anoxygenic phototrophs. And the shell proteins are homologous between cyanose and anoxygenic phototrophs. So AGT is feasible in this case. We have not found the donor or the microbe in between, but we recently started exploring another possibility, which is vertical evolution. As I mentioned before, there are some cyanos that do not do photosynthesis, and we actually found some bacterial microcompartment in these microbes, and series chroma share series. So we found the proteins. My student found them actually. Of course, it's not carbon fixation related. We found the rest of the genes in, so it's confirmed that the BMC is present. It's related to another type of metabolism, but it brings up the possibility that the BMC was in the last common ancestor of all cyanobacteria, including the non-photosynthetic ones. And then it acquired different functions in the two groups. So very quickly, we are trying to explore this question in more detail. However, we have serious suspicions that series are very biased in terms of their amino acid composition. So if you have more genomes for series, let me know, I'm looking for them. So very quickly, the conclusions is that as soon as oxygen become available, Rubisco needs something, right? In this case, the carboxysome was the solution. And I started thinking about whether this metabolism, well, this trait may have evolved in a densely packed community like a microbial map, or whether this is actually a universal feature of oxygenic photosynthesis. I will skip the rest to please look. I'm looking for a postdoc to apply to this fellowship and work in the lab, machine learning, protein evolution. So if you know someone, please send them my way. And with that, I thank you for your attention, and thank you to the Simons Foundation for supporting this work. Thank you.
A couple of things. One, are you familiar with carboxysomes across the bacteria other than in photosynthetic organisms like Salmonella uses them for detoxification?
Yes.
Yeah. Oh,
this is a bacterial microcompartment, not a carboxysome. So it's another type of.
Well, the Salmonella one is a carboxysome.
Oh, okay.
It's for detoxifying I think it's acetaldehyde.
Yeah, some BMC help with detoxification, but they are not involving carbon fixation.
No, it's not for carbon fixation.
Yeah.
It's common as a compartment for detoxification.
Yes, it is.
And then the question, you kept saying that the oxygen was low, and I'm just thinking about, heterocysts. A lot of the cyanobacteria make heterocysts. Is it cyanobacteria algae? Heterocysts because you can't fix nitrogen because they're making a ton of oxygen. So the idea that there's no oxygen around in an oxygenic species, I'm just wondering, to me, it would make sense they'd make them at the same time because otherwise you're generating the toxic compound that's going to affect the Rubisco.
Yeah, absolutely. That's why I say that it's an important mechanism for oxygenic photosynthesis itself. So I don't think it had much to do with the global conditions at the time, more like the metabolism itself.
Okay, let's thank the speaker again. Presentation by Dr. Graham from the University of Chicago.
Hello. Okay. Hi. Thanks for having me. My name's RJ Graham. I'm here to talk about potentially longer future lifespan for land plants on Earth than what previous studies have kind of suggested. So kind of have two pathways that are usually cited for the end of land plants on Earth. They both come from the fact that luminosity of the sun is increasing over time. So one of these pathways for land plant extinction is CO2 starvation, and the other is overheating. So as people have noted, Rubisco can have trouble discriminating between CO2 and O2, and it can run both ways. So when it photorespires, the net photosynthesis is less efficient, and as you increase temperature or decrease CO2, the photorespiration happens more, and so there's kind of a stress threshold where past a certain low CO2 or high temperature, land plants on Earth presumably won't be able to function because their photosynthesis won't work, assuming no, crazy biochemical innovations or evolution. So the sun, like I said, it's brightening over time, it's warming the Earth, and the silicate weathering feedback, which is part of Earth's carbon cycle, responds to that brightening by decreasing the atmospheric CO2. That happens because silicate weathering pulls CO2 out of the atmosphere. It's temperature dependent. It goes hotter. It goes faster when it's hotter. The sun brightens, you heat up the Earth. Silicate weathering speeds up and draws CO2 down until the net CO2 drawdown rate is in equilibrium with the input rate by background volcanic outgassing. And so on the left, that's just showing the increase in solar luminosity. On the right, that's showing the kind of estimates of decreasing CO2 through time from 4 billion years ago to today. So if you imagine continuing that into the future, that would be bad for plants. Similarly, at the same time that the CO2 is going down and the sun's brightening, Earth's temperature, like on average, will tend to increase in the future too. So the main objective here is to calculate how far in the future land plants and the life that depends on them will die due to either CO2 starvation or overheating in this framework. So there's an early kind of calculation that lots of people have kind of mimicked since then, Caldera and Casting 1992. I'm going to refer to it as CK92 throughout the rest of the talk because we use it as kind of a reference point. They found that plants, they expect them to die by CO2 starvation in roughly 1 billion years, basically. And the plots there are just showing time in the future, the evolution of CO2 in the atmosphere and the soil as the increasing solar luminosity happens. And then on the bottom there, you see the productivity that they use a simple representation of plant productivity as a response to CO2 and temperature. So similar results by quite a few other groups and people. They've done similar calculations. Not always, but typically they find that plants are expected to die by CO2 starvation. And when they actually do like the carbon cycle modeling, and I'll get into more detail about what those numbers mean, but when they do the carbon cycle modeling, they tend to find that it's going to happen about 1 billion years in the future. Okay, but most of those assumptions had kind of questionable modeling assumptions. So that equation right there is kind of just the standard picture that people use for continental silicate weathering, which again, that's that CO2 drawdown mechanism that feeds back onto Earth's climate and stabilizes it, like I mentioned. The W is weathering, V is outgassing, so you assume those are equal. The weathering rate is an exponential function of temperature, and it's a power law function of the CO2 in the soil. TE, that number that's in the red box, that's the basically controlling the strength of the temperature feedback of the carbon cycle. If TE is bigger, it takes a bigger change in temperature to cause a given change in weathering. If TE is smaller, you cause a big change in weathering with a small change in temperature. Previous efforts have typically assumed that TE is pretty small, roughly 13 Kelvin, but recent estimates from a variety of directions have typically found that TE for Earth, based on kind of lab data, observations of watersheds, inversion modeling based on carbon cycle proxy data, they tend to find that the feedback strength is actually quite a bit weaker than people thought. And remember, this is in an exponential temperature dependence, so you triple that e-folding temperature and it makes a huge difference for the kind of dynamics of the system. And then similarly, these previous calculations tend to assume that land plants will die off at 50 Celsius and they'll die off below 10 parts per million of CO2. C4 plants can get down below 10 parts per million of CO2. And there's this totally bizarre plant that grows in Yellowstone, Dicanthelium lanigosum. It's a panic grass is what they call it, I think. And when that grass, when its roots are infected by a particular fungus, and when that fungus is infected by a particular virus, the plant can withstand extremely high temperatures, up to 65 Celsius. They have run, they do, put it in, did an experiment exposing it to 65 Celsius for 14 hours at a time with a little bit cooling off for 8 hours, kind of day-night cycle thing. It was able to live for weeks without dying. And bizarrely enough, if you take that weird symbiont consortium and you infect other plants with it, they also gain this thermotolerance. They tried it with tomatoes. It's kind of crazy. So that is what I'm going to take as like the upper bound for plant growth, because we know they can live at that temperature under the right conditions. And then similarly, cacti, they can get really hot in their interior. So basically, land plants can go pretty hot. I think 50 Celsius was too conservative. 10 parts per million was too conservative. So what we did, we have a really simple global mean model with climate as a function of CO2, temperature, and sunlight. Outgassing is putting in CO2, silicate weathering is removing CO2, plant productivity is determined by temperature and CO2, and plant productivity feeds into silicate weathering by altering the soil CO2, because really the CO2 that's doing the weathering is in the soil, touching the water, and plants can increase that by productivity and then decay in the soils and things. So yeah, standard silicate weathering representation, kind of standard representation of how plant productivity affects the soil CO2 pressure. P soil is the soil CO2 pressure, and that's what feeds into this weathering calculation for the direct CO2 dependence. Then we calculated the net primary productivity with some simple but certainly questionable a pair of representations of C3 and C4 photosynthesis that kind of allow you to plug in CO2 and temperature, and under some assumptions about humidity and light and nutrients, you can get out productivity estimates from that. And so basically what we find is if you allow for more, 2 minutes, 3 minutes? Great, okay. If you allow this weaker temperature feedback that than people have previously used, and if you allow for the kind of hardier plant thresholds based on just the observations of existing plants on Earth, not even accounting for the possibility, the certainty that they will evolve a great deal in the next billion years, considering that C4 and CAM photosynthesis both arose in the last 30 million years, which is kind of crazy. Yeah, we find that when you account for that, the future lifespan is significantly longer, i.e. plants can last significantly longer, and also that instead of dying due to CO2 starvation, they tend to die by hitting that overheating threshold instead. So what I'm showing here, I'm comparing the calculations with the set of parameters that were used in kind of the standard previous calculations, CK92, that Caldera casting 92, and then the pink is with kind of updated parameters from a different paper. I'm labeling it KT17 here. It tried to estimate carbon cycle parameters based on the last 100 million years of geologic proxy data. And so you see that the top one is showing C3 and C4 for both of those scenarios. The C3 plants in both cases kind of monotonically decrease and die off after a few 100 million years. But the C4 plants, interestingly, in the updated parameter set, they maintain quite high productivity for a very long time before falling off. And that interacts in fun ways with the kind of evolution of the CO2, such that you, instead of getting that monotonic decrease, you get this kind of rolling trajectory, which prevents the plants from actually hitting their CO2 starvation threshold. So they stay alive until they hit the temperature threshold. Once you're in that overheating threshold, it turns out that outgassing's effect is much less important. So if you change the amount of CO2 outgassing, that'll tend, in the CO2 starvation regime, that's going to increase the outgassing. You're going to increase your lifespan, right? Because more outgassing means more CO2, means it takes longer to get down to that minimum threshold. Overheating, less so, okay? Another undergrad research did some calculations where she allowed the outgassing to vary stochastically, and she added seafloor weathering. Basic point there, stochastic outgassing under realistic assumptions doesn't make that much of a difference, and seafloor weathering could matter if it's very strong. It might shorten things and put you back into the CO2 starvation regime. Basic point though, if this point about weaker weathering and more resilient plants is true, then they might last, instead of about 1 billion years, they might last to up to 1.86 billion years into the future. If you decrease outgassing when you're in the overheating regime, then you're going to delay the time that you hit that temperature, and so you actually extend the future lifespan. And similarly, growing land fraction is the same thing in this very simple low-dimensional model. Both of those are expected to happen, decreased outgassing, growing land fraction in the far future. If that's true, then they may actually be able to exist on Earth until Earth is nearly in this moist greenhouse state where it gets really wet and everything, the planet starts losing water. If that's true, it may be relevant for biosignatures. That's my talk. Thank you. Oh, right, I guess I should keep that in case someone asks anything.
Thanks, that's really cool. I mean, we think about this and the shift back to an anoxic world when you don't have all the O2 production, and I never worry about this when I talk to my students. They say, we've got 1 billion years, don't sweat it. But I was just sitting here thinking, when will plate tectonics stop? And there's some estimates that will stop before this even happens based on your longer projections.
Yeah, that would be interesting. That would, plausibly kind of lead to a big decrease in your outgassing. And so if that were to be the case, exactly, yeah. So you could, well, yeah, exactly. It could go either way. You might actually end up having too little rock production to do the silicate weathering, and then you could allow CO2 to build up too much and then things die. Or it could be the other way. If outgassing slowed down a lot, then the CO2 might decrease and things might die from starvation earlier. So yeah, that's an interesting point. But I think basically the tectonics is so poorly understood that no one has any idea when it started or when it's going to end. So, but valid point, totally.
Okay, let's speak on that.
All right, thank you to all the speakers. This is really wonderful.
Okay, so we're going to go over the breakout sessions, and we'll be having a group photo afterwards. One thing that came up during yesterday's breakout discussions is that the idea of putting out a white paper was bringing up research ideas, and some of these haven't been done, and maybe they're cool ideas. So we want to just raise the point that any ideas that you learn about from this conference that could be research, be sure to follow our code of conduct, collaborate with the person who had that idea. Also, I think everybody agrees maybe it's better if we maybe do a publishable paper than just a white paper. So we're going to emphasize more writing a review paper, in which we don't have to present new ideas, but we will point out research gaps for this review paper. So that's anybody want to raise any other thoughts about that? Is that fine with everyone? Okay. Oh, all right, great. We'd like to hear now from the different groups, if you can, the designated note taker for those groups, so they could come up and go over some of the things that you guys discussed. Is there no one? Okay. All right. Which group are you in, Anastasia? Okay. Yes. Thank you. All right.
Yeah, thank you. There were other note takers,
so
please add
okay.
on. I did not
Okay.
cover everything.
All right.
I just have 2 notes that I summarized.
Okay. Thank you.
Hi everyone. So I was part of one of the first breakout groups with Avi, and there were 2 main, clusters that we saw that I put down. One of the first ones was that it would be great to have the connection between laboratory experiments, the evolutionary biology and planetary models that could be matched with some astronomical signals. And there are many of the participants repeatedly emphasized linking molecular and organism scale processes, such as photosynthesis, to the spectra, atmospheric signatures, and the telescope observables relevant to missions such as Habitable Worlds Observatory. And some of the specific items that were pointed out was what does photosynthesis look like under high CO2 and low or high light. I think the high CO2 we had the interesting talk earlier today by Bo, and then but also links to geology. And one of the other items that was pointed out would be the pre-photosynthesis conditions that could have been also observable. And then, if photosynthesis exists, how could it also look spectroscopically through different atmospheric conditions? And then the second point was expanding and diversifying the potential alternatives for life and biosignatures, such as what would spectra look for non-photosynthetic metabolism or extreme planetary environments, alternative biochemistries, and others. So those were the 2 clusters that I saw, but the other people that were involved in note-taking, feel free to add. Thank you.
Okay. I was also note-taking during that same session. I think most of the things were covered already, but I'm just going to scroll through really quick and make sure I cover everything. So I think we kept coming back to this point of what does photosynthesis look like as CO2 increases and stellar flux decreases in the outer habitable zone, and how does that manifest observationally? So going back to that idea of tying, ground truthing with Earth observations and modeling approaches with observational requirements for telescopes like HWO. So we definitely talked about the habitable zone quite a bit. We also started off the conversation with talking about whether we wanted, initially when we thought we were going to be doing a white paper, whether we wanted to focus more on the science or the advocacy angle. So really kind of whether we want to angle ourselves around telling NASA what types of things we need in order to encourage more discussions similar to what this workshop is enabling between different groups of scientists to make sure that we are addressing these types of research questions and also funding interdisciplinary work more, which is a really big, important thing for these types of questions that we're interested in. So advocacy, programmatic recommendations for NASA. There was also a mentioning of tying together NASA DARES, which is kind of under the Planetary Science Division with HWO CSIT operations, the Community Science and Instrumentation Team for HWO, which is more astrophysics division. So tying together those two divisions of NASA and utilizing like RCNs, so the research coordination networks that NASA is helping run to sort of try to initiate more workshops similar to this. I think that was pretty much everything, but yeah.
I was in no way prepared to share this list with people, and I probably shouldn't have been the designated note taker, but so yeah, our group was generally focused on pigments, questions around pigments that might be relevant to astrobiology, but even an understanding of the sort of physical basis of what could be possible on Earth. So a few of the ideas that we sort of came up with, something was brought up during the discussion yesterday where it was something like, can you have any kind of polymer, not only protein, but an example was given as RNA with folding that bound some kind of pigment. So that would be an interesting, even testable type of experiment, right? Using different molecules that we don't, we don't really think about on Earth. What is the chemical space for molecules that absorb similarly to chlorophyll? One of the points that came out of Jonathan Lindsay's talks yesterday was that there are some molecules that look spectrally very similar to chlorophyll molecules, and just kind of that sort of chemical space in general could be explored with different organic chemistries. Could there be a membraneless reaction center? Essentially the novelty of being able to energize electrons without thinking about the rest of metabolism. Could that be a useful thing outside of the metabolism we're used to thinking about? Pigments could be involved not in light harvesting, but perhaps only for photoprotection, which I don't know how much you see of. Certainly like not in photosynthetic organisms, right? And then we have examples of chlorophylls in the context of our biology, and we have people who tend to study chlorophyll biosynthesis and things like that, but we don't really have the organic chemistry expertise involved in a lot of those conversations to understand the simplicity of the chemistry that's involved in making those things, right? And that simplicity may be very different from how life has evolved to actually synthesize those pigments. So that's kind of a gap that came up. Anybody else from this working group want to share anything that? Okay, I also recorded it, so yeah. You need this.
We didn't have time to talk about that HET stuff, so I don't know, maybe it could be interesting.
Can you repeat it?
Yeah, we didn't have time to go through that HET, like the transfer of genes that might be important for photosynthesis in general. So I don't know if that's something we want to revisit maybe later. Or in general, evolutionary events that might be important for photosynthesis.
Okay.
Okay, there were a couple of us taking notes for the breakout group on water oxidation. I'll share my notes. Nikki, do you want to share some of your notes? Or another. Oh, you did. Okay, why don't you come up and share your notes?
Sure. Thanks. Yeah, so we had some overlap with the first group in that we also were discussing essentially what conditions did photosynthesis evolve under and what conditions is photosynthesis capable of tolerating, although kind of more on the side of evolution of oxygenic photosynthesis. We discussed, is evolution of the oxygen evolving complex inevitable? What is the probability it would happen? What intermediates could happen? Can there be less efficient versions that perhaps can't go all the way to oxygen evolution, but can be part of a pathway that involves superoxide dismutase or peroxide metabolism as well? Can anything other than manganese be used in a practical basis? Let's see, could we use a snowball Earth type condition as a means of detecting that as a means of detecting oxygen? Let's see, we also talked about using nitrogen redox metabolism as a component of this. And I'm guessing you have more notes than that as well.
Okay, yeah, so a lot of it was how do we get toward water oxidation first on Earth and then also in other planetary contexts. So they're definitely intermediate pathways in this idea that that Usha brought up, that manganese is an antioxidant, could have just been a precursor that then enabled the OEC later. I could think RJ brought up this idea of we need to kind of explore, this could be really laborious, but explore the potentially inefficient space in different planetary contexts to evolve some cofactor capable of water oxidation. Actually, used poster is already actually trying some of that, but I think the talks have pointed out that nothing is better than manganese at this point, but we need to look at this in other environments. I was asking Junko also earlier, has anybody subjected the OEC to just UV radiation? Because that's an environment where it would have had to have emerged in a high UV environment initially. So let's see, for snowball states, producing oxidizing species, nobody had quite expertise about that possibly, but these could have produced precursors also, the OEC. That's what I have so far. Yes, Hazel.
Did you consider the energetics of the electrons going into the OEC? Like just thinking if you have lower energy electrons from like
I was talking about that.
Yeah.
I wasn't sure how to make this something operationalizable, but I was noticing that the reason it's like all the anoxygenic phototropes are hooked up, the reaction centers hooked up to other complex chains and other enzymes, whereas the business end of the reaction happens inside photosystem 2, and that's because there's no redox cofactor or no redox intermediate that's oxidizing enough to mess with oxygen. We were wondering if that was a thing that was possible in another biochemistry and we just got unlucky, such that you needed this fancy, weird thing, or if it would be possible for another redox cofactor to be like that.
So thank you all for those notes. I think all these topics in the different breakout groups were far too large for us to cover, and of course we couldn't cover everything. So what I've also made some extra folder in our presentation or presenters folder where you put your presentations. So this is the folder where the breakout groups, you can put your notes to share with each other. So for moving forward for this review paper, since we're all, I don't know yet if we should do one review paper or multiple. So
I'm thinking when we come back from lunch for the working groups, we maybe we should start it with one plenary working group, and we will flesh out an outline altogether. I'll make a Google Doc, and everybody, you can jump in and we will start writing into it and do an outline. Then we'll see how that evolves into maybe breaking it onto more than one paper. And then we'll see who's going to take leads on different sections. Anybody, Bob, you just shook your head. Is that a bad idea? You've done this before. Okay, been there, done that. I think it works. It's amazing how many people will jump in and write in Google. Oh, okay, Bob doesn't like Google Docs, but okay, rest of us do. So anybody have other approaches you want to suggest before we plow into that approach?
I have a question. Do you guys want more discussion time to
Yeah.
flesh out these topics?
Yeah. Good. I was thinking maybe there are post-lunch work here will be a discussion time but we can write at the same time that we're doing discussion. Does that sound good?
Discussion of all of us, not in breakouts.
Right, not in breakouts. We'll be all together in one, because I think we have so much expertise that's diverse, and it's really hard breakouts. When we wanted somebody, it's like, oh, they're over in that group. So, okay, we are right on time for the group photo. So we're gonna go out this door, down the stairs. No, you don't have to wear. You can wear what you want. And so we have a yeah. All right, all of us down. We'll try to get the link.